So today we are going to talk about carbohydrates. Carbohydrates are sugars or saccharides, and they're organized in a number of ways. We are going to look at 3 ways in which carbohydrates can be organized. First, they can be organized depending upon the type of carbonyl group present. For example, there are 2 types of carbonyl groups, aldehydes and ketones. You studied that in your AP courses of chemistry. For students of India and Pakistan, AP is college chemistry or intermediate chemistry that you study in grade 11th and 12th. If an aldehyde is present in a sugar, it's going to be called an aldose, and if a ketone group is present, it's going to be called a ketose. So depending upon the type of carbonyl group present, we have 2 types of sugars, aldoses and ketoses. A second way to organize carbohydrates is on the basis of the number of carbon atoms present. For example, if there are 3 carbon atoms, we are going to call that sugar a triose. If there are 5 carbon atoms, we are going to call it a pentose. If there are 6 carbon atoms, we are going to call that sugar a hexose. Examples of these are glucose and fructose. Glucose is a 6 carbon sugar, that's why glucose is a hexose. And fructose is a 5 carbon sugar, that's why fructose is a pentose. Also, reflecting back on the first way of organization depending upon the carbonyl group, glucose is an aldose. The classification of carbohydrates determine the number of sugar molecules present in the carbohydrate. For example, if you have just 1 sugar molecule, it's going to be called a monosaccharide. The most primary unit of sugar is glucose. Glucose is a monosaccharide, and glucose is the fundamental monomer of carbohydrates. So before we delve further into classifications, let's talk about glucose. Glucose is a 6 carbon sugar, it's an aldose, and it's a hexose. What you need to understand is you need to look at the structure of glucose, and you need to look at different types of structures present in the book. Usually there is a straight chain structure, and then there's the cyclic structure. Also, you need to look at Fisher projection and the chair projection or board projection. It is important to note that sugars containing 5 carbons or more are rarely seen as open chain compounds. So glucose is usually represented as a cyclic molecule, which means it is shown as a closed ring. So when a glucose forms a ring structure, the carbon present on the carbonyl group is called the anomeric carbon, right? So if you remember isomerism from your AP course of chemistry, isomerism is a phenomena in which a substance has a same chemical formula but a different structural arrangement. All right, so you might have 2 substances that share the same chemical formula, the same number of atoms, but how those atoms are arranged might be different. If you cannot recall isomerism, I would like you to go back and revise your AP courses. We are going to study 3 types of isomerism that are commonly observed in glucose. So depending upon the anomeric carbon present in a ring structure of glucose, there are 2 anomers of glucose, that is the alpha glucose and the beta glucose. Read up on those 2 structures. They are very simple. It's just a difference of a hydroxyl group. It could be up, it could be down. All right. Also, there are enantiomers. Enantiomers are basically optical isomers. Optical isomers means that they are mirror images. You have 2 compounds, and they're optically identical, which means that they look same, but if you try and superimpose them on top of each other, you are going to find out that they do not match. The best way to understand optical isomers is by your hands. See, your hands are basically mirror images of each other. So they look same, but they're different. And how would you know that they're different? When you try to place your hands on top of each other, they don't match, right? So optical isomers of glucose are basically represented as L and D isomers, or left-handed or right-handed isomers. The L and D represents lever rotatory or dexter rotatory. You need to go and read upon this further in order to understand it better. And then the third type of isomer that is commonly observed in monomers or monosaccharides is called the epimer. So epimers are basically substances that are different around one carbon atom. There are two epimers of glucose. One is different at carbon 2 and the other is different at carbon 4. Alright, so now that we've discussed about anomers, enantiomers, and epimers, we are going to discuss further classification based upon the type of sugar units involved. Since glucose is a monomer, we are going to now go towards a disaccharide. Alright, so disaccharides are basically two sugars joined together. What you need to understand is that the bond that joins two saccharides is called a glycosidic linkage or a glycosidic linkage. And there are two types of glycosidic linkages. One is called the N linkage and the other is called the O linkage. The N linkage consists of a sugar molecule being attached to an amide group, which contains a nitrogen, whereas the O linkage contains the sugar being attached to a hydroxyl group. The important part of disaccharides is that you need to memorize some of the most common disaccharides, and that's just it. Remember that two sugars joined together in a glycosidic linkage is a disaccharide. Most common disaccharides, you can find them in the book. There are 3 to 4 disaccharides that come up in the exam continuously. Moving on further, we have another category called oligosaccharides. Depending on your book, oligosaccharides are classified as saccharides containing 3 to 12 or 15 sugar units, sugar monomers. Alright, so if you have a carbohydrate that is made up from 3 to 12 or 3 to 15 monosaccharide, it's going to be called as an oligosaccharide. Alright, next we have a polysaccharide. A polysaccharide, as the name suggests, contains sugar units which are greater than 12 or 15. So our most common polysaccharides are glycogen and starch. Both of these are storage forms of carbohydrates. Glycogen is found in animals and starch is found in plants. However, the glycosidic linkage between their monomers is different. It is important for you to understand the structure of starch and glycogen. So a glycogen molecule consists of a straight chain and a branch chain. Alright, so there is a straight chain of glucose units, and then there are branches emitting out of straight chain. Now this straight chain is made up of glycosidic linkage, which is formed between carbon 1 and carbon 4, but those branches are made between carbon 1 and carbon 6 of the same glucose. Alright, so that's how glycosidic linkage is different depending upon the type of polysaccharide under consideration. I want you to go and look up at the structure of starch and explore how it's different from glycogen. Usually you also need to understand different tests you should do to determine different types of carbohydrates present. Mostly these topics are covered by your lab, and it is important both clinically and in terms of understanding that you need to understand how to detect different types of sugar molecules. At this point, you also need to understand that some sugars are reducing sugars, whereas other sugars are non-reducing sugars. So basically, reducing sugars are sugars that when they react to a substance, they lose their ringed formation because the carbonyl group reacts with the substance and gives a colored output. This is important for you to remember. All monosaccharides are reducing sugars. All disaccharides are not reducing sugars. There are some disaccharides that are reducing sugars. fructose is also reducing sugar. Polysaccharides are further classified into two groups: homopolysaccharides or heteropolysaccharides, depending upon the type of subunits that it contains. If a polysaccharide is made up of similar sugars, similar subunits, similar monomers, for example glycogen, it's called a homopolysaccharide. If a polysaccharide is made up of different monomers, it's called a heteropolysaccharide, which is the case in terms of starch. Carbohydrates make substances called GAGs. GAGs are basically long chains of heteropolysaccharides which are negatively charged, and they accumulate water around them, giving them a spongy texture. GAGs are present in our synovial fluid, they're present in the vitreous humor of our eye, and they form the extracellular matrix of the cell. Now the extracellular matrix, or the ECM, is basically a cytoskeleton, which is the skeleton of the cell. It gives the cell its structural integrity. So if you haven't studied cell biology, let me tell you that there isn't just cytoplasm inside the plasma membrane. There are tiny microscopic thread-like structures which are called fibrils, I think so they're called fibrils, and along with these GAGs, they provide a skeleton to keep the cell in its shape. Apart from this, there are two other molecules, proteoglycans and glycoproteins. Once again, glycoproteins, proteoglycans, and GAGs are separate topics that I will cover in another video. But for now, you need to understand how to differentiate between proteoglycans and glycoproteins. Focus on the name at the end. When I talk about proteoglycan, the glycan means that it's a sugar, it's a carbohydrate wrapped around a protein core. Alright? So an example of a proteoglycan is GAG. There are different types of GAGs, and these GAGs are proteoglycans. Whereas when you talk about glycoproteins are basically protein that have a small amount of carbohydrate. When proteins are made, they can be modified to add a piece of sugar to them, making them glycoproteins. And glycoproteins are mostly found on cell membranes, and they serve as cell signaling molecules, right? They tell other cells what type of cell they are. They basically help in recognition of a cell, alright? It is important for you to understand what are glycoproteins or what are proteoglycans. Keep your concepts clear because it is clinically important as well. Not going into details, but if you have studied microbiology, certain bacteria have certain sugars outside their membranes. Alright? Just giving you a hint. If you want to read, you can read further on it. Also, your blood group is determined by the type of glycoprotein present on your red blood cell. You should also read on that. It's a very interesting topic. So glycoproteins, proteoglycans, GAGs, polysaccharides, monosaccharides, disaccharides, types of isomerisms, types of structures of glucose, covers your entire topic of carbohydrate. I hope that I was clear enough to help you summarize it, and I hope that this video helps you revise the topic quickly. If you have any confusions, you can write down in comment, or you can send an email, or you can reach out to me on Twitter, and I'll be glad to answer your questions.